Okay, so good evening to one and all present here. Okay, so I welcome you all to the week 4 live session on the course Experimental Biochemistry. I am Srijan Sen from IIT Kharagpur, and today I will be taking your week 4 live session on this particular course. So I'm sharing my slides with you all. Yeah, so I guess I am visible and audible to you all. So if that's fine, then we should proceed with the lecture. Great. Okay. So so today's live session. So till now, how much we have spoken about is basically on the very basics of biochemistry, and it had a lot more of the concepts that are related to biochemistry than it is directly involved in biochemistry. Okay, so like we have spoken about the errors and the precision, accuracy. Then we moved ahead with discussing about the amino acids, peptides, and all. Then in the last class, we spoke about the spectroscopic techniques and the details about it. So these are a lot more of chemistry than biology. Okay, so here we will be, in today's lecture, we would be dwelling more into the biological system, that is about proteins. So I would be introducing to you what proteins actually are. Most of you definitely know about proteins and have studied about proteins, but still I would like to introduce you to proteins and how we can get these proteins, what's the role of proteins, and other applications of proteins and the related informations about it. Okay, so when someone around us asks us what proteins are, it's not like it's a biological molecule that has different functions in our body, but if we talk from a scientific or a biological point of view, proteins are biopolymers that are made up of 20 different L-alpha amino acids linked via the peptide bonds. So there are the peptide bonds that are responsible for the linkage between the different amino acids or the peptides, and uh, sorry, wait. Between the amino acids or the peptides, and this combination of the peptides lead to the formation of large biomolecules or biopolymers known as proteins. Now the polypeptide backbone of this protein is a repeating sequence of NCC, because we have the N terminus and the C terminus and an alpha carbon that forms the backbone of these proteins. So in the N terminus we have the amino group, in the C terminus we have the carboxylic group. Now apart from this we have the C alpha in between the N and the C terminus, and the C alpha is attached to an R group. The R group is responsible for providing D alpha amino acids. You do not usually naturally get a D alpha amino acid. Is it so? Because you find D alpha amino acids in only some specific, like the bacteria can produce this D alpha amino acids. But what about, it's not naturally occurring. We cannot call D alpha. So in our body, usually all are yes, all are L alpha amino acids only. We do not find the D alpha amino acids in the body. Like for humans, I'm not sure we get D alpha amino acids. So we cannot call it to be naturally occurring. So the L isomer of amino acids are more prevalent. Okay. So the side chain or that is attached to the C alpha group is basically the, so varying this R group is basically responsible for the formation of the 20 different amino acids, right? Now, as I was speaking, it's linked by a peptide bond. So how are these peptide bonds formed? So peptide bonds are formed when there are, so we have two amino acids, say amino acid X and amino acid Y. And then what happens, the OH of the carboxylic group and the H of the NH2 group. So this dehydration reaction occurs via the removal of one molecule of hydrogen, one molecule of water, via the formation of a peptide bond. So this is the peptide bond. You have the CN, that's the peptide bond over here, that is involved in linking the two amino acids via dehydration reaction. So how do you know the OH is lost from the carboxylic group and the H is lost from the NH2 group? Now, if you see the examples of which are given over here about the polypeptide chain formation, where you have the number of peptide bonds are equivalent to the number of molecules of water lost. So if one molecule of water is lost in the formation of the peptide bond, that means there are two amino acids that are involved, and there is the formation of only one peptide bond. But this is only the case if it is a linear peptide. But however, if there is a cyclic peptide, like you are given that there are 7 hydrogen water molecules lost and there are 7 amino acids, how is that possible? Because in a linear peptide there are supposed to be 8 amino acids with 7 peptide bonds with 7 molecules of water lost. But for a cyclic peptide, what happens? It's a circular molecule. So the first amino acid and say the 8th amino acid, they come close to one another via the formation of their peptide bonds. You get the equivalent number of peptide bonds. So you have 8 amino acids, 8 peptide bonds, and 8 molecules of water lost. So this is only the scenario for a linear peptide. But for a cyclic peptide, one additional peptide bond generation is arises along with the one molecule of water lost. Now, the peptide bond, we recognize these peptide bonds via a few characteristics. So these peptide bonds are the strong bonds and they are covalent in nature. Now, they are covalent in nature, followed by they have a partial double bond character. Like neither they are single bonded nor they are double bonded, but they have a partial double bond character. How we can understand that? Because the distance, when we measure the distance between these peptide bonds, it is more than that of a single bond, and it is less than the it is less than that of a single bond and more than that of a double bond, and its approximate distance between the C and the N is about 1.32 angstrom. Second, thirdly, it has it's rigid and it gives planar structure to these amino acids. Okay, followed by the trans configuration of the alpha carbon atoms. The side, because of its proper orientation to interact with the other, with its neighboring amino acid. And also the side, these peptide bonds are so rigid and planar that it allows the side chains to freely rotate on either of these peptide bonds. Now, this is the main bond that is actually responsible, or that plays a vital role in the okay. So I just missed your question. So it's the bond formation in the case of a circular amino acid. Okay, so for example, what you see over here, you have a methionine, aspartic acid, leucine, and tyrosine. So here you have 4 amino acids, right? So for 4 amino acids, how many peptide bonds are you supposed to have? So you are supposed to have one over here, 1 bond, 2 bonds, and 3 bonds. Okay, so 3 peptide bonds with 4 amino acids. So 4 amino acids forming 3 peptide bonds for a linear peptide, or a linear, yes, for a linear peptide. But if it is a cyclic peptide, what does it mean? For a cyclic peptide means it does not have open N and C terminus. It is only possible when the methionine NH2 or the NH3 plus forms a bond with the tyrosine carboxylic OH group. Okay, so the OH and the H from over here is lost and the bond forms in between. It is only possible when the orientation is supposedly like that. Okay, like here we are seeing a 2D diagram, but in 3D it's very close to one another. It can be, so a bond formation is possible. If that is done, then you have 4 amino acids, 4 peptide bonds, means 4 molecules of water are lost. That means this is a cyclic peptide. Okay, but in this case I am showing a linear peptide, not the cyclic peptide. I guess that is clear to you. Yeah. Okay, so these are the characteristics of the peptide bond. Now, apart from the peptide bond, now peptide bond plays a very vital role in the backbone structure of a protein molecule, because amino acids are connected via peptide bonds, right? Great. Now what do you see over here? There are other bonds or other interactions that also play a vital role in the structure, function, as well as the dynamics of a protein molecule. These are the three very important terms which you will definitely see in terms of a protein molecule, like structure of a protein, function of a protein, or and the dynamics of a protein. These three are very, very important terms. So we have the strongest of the bonds known as the covalent bonds in the form of peptide bond as well as the disulfide bond. So how are these disulfide bonds formed? From the name, if you can understand, there are two sulfur atoms or the sulfide groups that are involved in the formation of a bond. So you have the SH group. You have two SH groups that are forming the disulfide bond. So one amino acid that forms these disulfide bonds is the cysteine. So cysteine forms the disulfide bonds that are involved in a stable structure of a protein, the proper protein folding. So protein folding via the disulfide bonds gives a you can tell a stable structure to a protein molecule. Now, other non-covalent bonds or interactions includes your hydrogen bond, ionic bond, and the hydrophobic interactions. So disulfide bonds form between the two cysteine molecules over here. So what you have over here, you have the cysteine molecule, you have the CH2SH group over here, and here you have the CH2SH. And this is in the reduced state. This is the oxidized state condition where the hydrogen atoms are lost. So you have the oxidation reaction and you get the disulfide bond formation. Okay, so if you can see over here, you see the disulfide bonds are very important in giving a structure. Otherwise, what would have happened? It would have been more of a disordered region, or you call the intrinsic, you might have heard about intrinsically disordered proteins. So these are the disordered regions. So these disulfide bonds play a very important role in giving a proper orientation or structure to a particular protein. One example of such disulfide bonded protein or sequence is your bovine insulin. So insulin, if you see over here, you have intramolecular as well as intrachain as well as intrachain disulfide bonds. So there are two chains present over here. You have the chain A and the chain B. So chain A is connected to chain B via two disulfide bond formation. Yes, it is because of the methionine does not have it, because if you look into the structure of methionine, it has CH3. It does not have a free H group that can undergo this redox reaction. Okay, the CH3 entirely is not lost. That is why it does not form a disulfide bond. Now, so chain A is forming disulfide bond with chain B, two disulfide bonds, and there is an intra chain there is one intra chain disulfide bond in this insulin molecule. Now, hydrogen bonds are formed between the positively charged hydrogen atom and the negatively charged O. So, oxygen. So, we have these hydrogen bonds are very important in the formation of beta sheets. So, I would be discussing about this later on. Like you have the alpha helix, beta sheet. So, these hydrogen bonds are very important in the sheets, and otherwise there are so many hydrogen bonds, or we have the hydrogen bonds forming between the negatively charged atom and the positively charged, or partially negatively charged atom and a partially positively charged atom. Right. Now, ionic bond formation happens between an negatively charged amino acid and a positively charged amino acid. We also know ionic bond formations as the salt bridge formation, because it forms a bridge-like structure where there is a charge. Uh, the charge is involved in the formation of this bridge, salt bridge formation. Like you have the arginine, that is a basic residue, and it forms a very nice hydrogen bond with acidically charged amino acids, like you have the glutamic acid, aspartic acid, and so on. Now, also now it is always not important that a arginine present in a protein molecule will form a hydrogen bond with a glutamic acid, or vice versa. Same goes with an lysine and an aspartic acid. Okay, the pH of the solution, the pH of the environment, the pH of the buffer in which your protein lies or your protein is stable will also affect the charge on this different amino acids, and those are also very important in the formation of the ionic bonds. Now, that's about the salt bridge. Now we have another very important interaction, that's the hydrophobic interactions that are involved in the formation of the protein structure as well. Now, what does it do? Now, we know hydrophobic molecules are those that are not water loving. So what they would like to do, now protein is present in a water environment. We talk about the environment and the core. So the environment is filled with water, where the hydrophilic molecules try to take the outer atmosphere and try to be in the outer environment, where they like to interact with the water molecules via the hydrogen bonds. But hydrophobic molecules, what happens is that they are not water loving. So they try to dig themselves into the core of a protein molecule. Like you have the outer, so there are two cores. One is the outer layer and one is the inner layer. The inner layer is mostly occupied by the hydrophobic molecules that forms the green portion, that is the core of a protein structure, and the outer molecules, that is formed by the hydrophilic molecules or the polar molecules, and they love to interact with the surrounding water molecules. Therefore, the hydrophobic molecules forms the core, and they are involved in the folded conformation in an aqueous environment, leading to a stable protein structure formation. So in summary, there are so many interactions and covalent bonds that are involved in the structure determination of a protein molecule. Like you have the hydrogen bonds, you have the hydrophobic interactions, the disulfide bridges, salt bridge formation, and so on. And these interactions are involved in the tertiary structure formation of a protein molecule. Now, a structure of a protein molecule. When we speak of a structure of a protein molecule, we can segregate these structures into different categories, like the primary structure, secondary structure, tertiary structure, and the quaternary structure. And these are the important parameters that also help us to differentiate between a polypeptide as well as a protein. You can tell what is a polypeptide, because this is a very general question that anyone might ask you, the difference between a polypeptide and a protein molecule. So a polypeptide is one that is a combination of a different amino acids joined via peptide bonds. It is not always important that a polypeptide is shorter in nature and the protein is longer in nature. You can have even an 80 amino acid long polypeptide. That is not a protein, because there comes the importance of a secondary structure or a tertiary structure. A protein molecule is a biopolymer formed via the L-alpha amino acid formation with a very folded structure having a definite structure as well as function. But for a polypeptide, it is not always important that you have a definite structure or a function. It can be a primary structure joined by a peptide bond. So anything that is joined by a peptide bond, we call it a peptide. Or if it's more than two or multiple amino acids joined, we call it a polypeptide. Now you were asking me why the D alpha amino acids are not there. So peptides or protein molecules are usually formed of L alpha amino acids. Probably if you want to introduce a an unnatural amino acid which is a D alpha amino acid, probably you won't call it a protein because it will not be involved in the proper structure of the protein. And this definite structure of each of these amino acids are also involved because they form the primary backbone, primary structure of a protein, and these are also involved for the higher structure formation. Therefore, a proper orientation of the amino acids as well as the you'd say the isomerism, the stoichiometry is also very important. So what is the primary structure? The linear sequence of amino acids forming the backbone of a protein. So you have the backbone of a protein, that is the formation of a primary structure. Starting from the N terminus, you have the N terminus which is your glycine as of now, and the C terminus which is your asparagine residue. Now, peptides can be dipeptide, tripeptide, tetrapeptides, and so on and so forth. So, oligopeptides can be more than 20 amino acids, polypeptides can be 20 to 50 amino acids, and your proteins are more than 50 amino acids. But proteins are the ones that should have a stable structure function dynamics. Now, what happens if there is a change in a single amino acid in the polypeptide chain? So what happens over here is that a single amino acid changes, and you see that a normal beta globin forms a mutant beta globin. So your hemoglobin, so your normal red blood cells changes to a sickle red blood cell, leading to sickle cell anemia. So a change in one amino acid is basically leading to a disease condition from a normal cell to a sickle cell anemic patient, right? But if you see over here, it is not the change of only one amino acid, rather it is only the change of one nucleotide. So your T is just changed to adenine. So your um, T changes to adenine, T changes to A, and you see a disease condition. Therefore, one nucleotide change or a single nucleotide, single nuclear polymorphism SNPs can also lead to disease condition. And one nucleotide change leads to change in the amino acid because it is replicated in terms of triplets. So three nucleotides lead to the formation of one protein. So therefore, it can lead to a disease condition. Now, so that's about the primary structure. The secondary structure is the regular arrangements in space of adjacent amino acid residues in a polypeptide chain. Like there are three types of secondary structures that can be possible in case of a protein. Like you have the alpha helix, beta sheet, beta turn. So helix, sheets, turns. Now, what are alpha helices? So, alpha helices are a result of hydrogen bonds formed between the carboxylic oxygen. So, you have the oxygen over here and the amide hydrogen of the polypeptide chain. And it does not depend on the side chain materials. Okay, so the carbonyl oxygen of each peptide bond is linked to a hydrogen bond with an amide hydrogen of the fourth amino acid towards the C terminus. So what do I mean by this statement? So you have an ith amino acid that the oxygen of the oxygen of the carboxylic group of the ith amino acid interacts with the hydrogen atom of the amino group of the I plus fourth amino acid. Like for the first case, if it is so, so you have 1, 2, 3, 4, and 5, and it goes on. So the first, so amino acid 1, the oxygen of the carboxylic group of amino acid 1 interacts with the NH, the hydrogen of the NH group of the amino acid number 5. And if you see the pitch, so what is the pitch? The difference between the one helix and the other helix, the turn of these helixes. So the pitch is about 3.6 residues per turn. So what are the features of these alpha helices? If you say it's more stable, it's more common and forms a stable conformation. The spiral structure, that's the polypeptide bonds form the backbone of the spiral groups. The R groups of the amino acid remains outwards and are not involved in the alpha helix formation. And alpha helices are stabilized by hydrogen bonds, and hydrogen bonds are weak, but they collectively determine the stability of the alpha helices. However, however, okay. So your question is, how does the pitch of a it is 3.6. Okay. So if you see over here, like the number of amino acids that are, so it's like about measuring the number of amino acids present in a turn region. Okay, so 3.6 indicates that there are 1, 2, and 3, and the fourth is not directly involved over here. So there is a calculation for it where you have that. So if I'm not wrong, it's like 36 residues, or um, there are something about 36 residues leading to the formation of 10 turns. So this was calculated in, sorry, I did not get your point. Uh, so if you see from here, there are 3 amino acids. Okay, great. Okay, so let me try to explain. So for, if you consider the scenario for a right-handed helix, or for any helix, if you check it, if it's a right-handed helix, it's consists for each residue, there is a rise of 1.5 angstrom. Okay, so the rise for every amino acid is 1.5 angstrom, and the total so then the pitch or the loop region, what happens over here is basically, so the distance between uh, the starting region of the loop and the ending region of the loop is 5.4 angstrom. No, no, no. It's not like that. It's not plus zero point 0.3 or something. It's like for every rise in the amino acid, for every rise in the residue, it is 1.5 angstrom. So one residue leads to a rise of 1.5 angstrom. Okay, in the amino, in a alpha helix, and the starting region of the loop to the where the loop ends, and then the next loop can start. So the distance between the starting portion and the end portion, that we call as the pitch. So the pitch of this alpha helix is 5.4 angstrom. Okay, and with every residue leading to a rise of 1.5 angstrom, so 5.4 divided by 1.5, I guess that would be equal to your 3.6 residues per turn. So one turn having 5.4 angstrom. So the distance is 5.4 angstrom. That is true for right-handed alpha helix. And for every amino acid, the rise is 1.5 angstrom. So that is equal to 3.6 residues per turn. Yeah. Okay. Now we have we have seen how these alpha helices form, but there are many factors that lead to the disruption of the alpha helix. Now, all amino acids are not involved in the alpha helix formation. They are known as helix breakers, and we have some amino acids that are helix formers. So among these, the helix breaker residues involves your proline and glycine. Why? Because proline having a very rigid structure, it is very difficult for a proline molecule to undergo such a sharp turn and lead to the formation of an alpha helix. Therefore, such rigid bond formations or such rigid structure makes it difficult for an alpha helix to form. Secondly, glycine being extra flexible enough with having super dynamics or being highly dynamic in nature is also attains a lot of orientations, making it also fall in the category of a helix breaker. Now, crowding of equally charged radicals, for example, too many positively charged or too many negatively charged molecules can also disrupt an alpha helix. Overcrowding of bulk R groups, like you have large R groups, side chains, though they are not involved in directly in the alpha helix formation, yet they can disrupt it, because there can be um, steric hindrances across each other, and that can also disrupt a alpha helix. Also, if there are certain chemicals that tend to form hydrogen bonds with each other, that can also lead to the disruption of this alpha helices. So we need to keep in mind as to about the helix breakers, like which residues are not in, should not be there, as well as what are the properties under which alpha helix formation gets disrupted. Now coming to the beta pleated sheets. So we have the anti-parallel beta sheets and the parallel beta sheets. So parallel beta sheets are the ones, so if you see over here, you have the parallel beta sheets where from it runs from the C terminus to the N terminus and the C terminus to the N terminus. Okay, so C to N, C to N. But for an anti-parallel, you have N to C and C to N. So they are already, the two strands are running anti-parallel to each other. And why is it more stable? Because if you see over here, there is not much, you can tell, a rough handling of the bonds over here, because they are positioned. So what do I mean by glycine is dynamic is that because glycine is so flexible that making it stay in one place is very difficult, because it's the smallest molecule, and as well as it does not want to be in a position where you can orient it in a particular direction. Okay, because it is flexible in nature. It's small as well as it's so flexible. So flexible, I mean by it's dynamically or entropically very so its entropy is very high, and it moves about, it's dynamic, it's more dynamic in nature. Now now both the models can be found in the proteins, but the antiparallel structure is more stable, that I was explaining. Like why? Because if you see the bonds, that these bonds also so the hydrogen and the oxygen over here are at very close proximity, so the bond formation is like the flexibility is the major issue over here. The size can vary, but it is the smallest amino acid. But the flexibility is a concern, because if it is too flexible, then the formation of the alpha helix gets hampered, because these are secondary stable structures that adds to the stability of a protein. If you have an amino acid that is more flexible, that can add to your loop region, that can add to other portions of the secondary structure. But where you have, where you need to form a helix, where or where there are some conformational strains, you need something that is exactly of that particular, that has that particular property to form the alpha helix. Like it is similarly for proline, you cannot have it because it's extremely rigid, so the bend around this region, so the bend around this region is not possible. As well as for a glycine, if there is a, it's so flexible, if it leads to a breakage or the loop is not complete over here, then it would not complete up entire turn, and that will actually hamper the alpha helix formation. That is what do I mean by an helix breaker. But it is always not important that your glycine cannot be in an alpha helix. There are many proteins where glycines are there, but that is for a that's because of they are naturally occurring, and the protein has a particular role of that glycine in its function. That is why it is there. So small does not matter, but the flexibility does matter for the formation of a alpha helix. Now, the other form of secondary structure is your beta turns, that permits the change in the direction of the peptide chain to get a folded structure. Like your prolines and glycines are very much prevalent over in this turn regions, and the beta loop the beta turns allow the protein compaction, since the hydrophobic amino acids tend to be in the interior and form the core, whereas the hydrophilic residues interacts with the aqueous environment. These turns are very important for this particular reason. Okay, so these three types of secondary structures form your are that forms the different portions of your protein. Now coming to the tertiary structure of a protein, so it gives you the complete information of a protein. So when we talk about the conformation of a protein, we refer to as the complete three-dimensional shape of the protein, which is comprised of your beta sheets, alpha helix, turns, loops, etc. So the tertiary structure represents the spatial relationship of the different secondary structures to one another within a polypeptide chain, and how these secondary structures are arranged in a manner and to form a three-dimensional form of a protein. So the tertiary structure describes the relationship of different domains to one another within a protein. Now, what is a domain? A protein domain is the basic structural unit within a protein molecule. So it is the part of the protein that can fold into a stable structure independently. Now, different domains together can form a protein. Like you have the beta alpha beta, you have the alpha beta beta, you have the beta hairpin motif, beta hairpin motif, and the domains over here. So there is a so these are the different domains that are maintained across the different heredity of the proteins. Proteins can have many more domains, that depending on the size, and these domains, multiple domains can lead to the formation of a particular tertiary structure of a protein. Now, this tertiary structure is based on the various types of interactions. So, these interactions are very similar to the interactions that maintain the structure of a protein. You have the salt bridges, the hydrophilic interactions with the water, the hydrogen bond, you have the internal hydrophobic interaction, the hydrogen bonds between the beta pleated sheets, the disulfide bonds, and so on. Now, the stabilizing interactions, so these are the stabilizing interactions in the tertiary structures. Like the hydrophobic interactions are the attractions between the non-polar alkyl and aromatic groups from, form a non-polar center that is repelled by water. That is why they form the core of a protein molecule. The hydrophobic interactions are the attractions between the polar or the ionized R groups and the water on the surface of the tertiary molecule. The salt bridge formed between the charged residues, the ionic interactions between the ionized R groups of acidic and basic amino acids. Hydrogen bonds occur between the polar groups of the amino acids, and the disulfide bonds. See, so let me complete it. So the disulfide bonds are the strong covalent links between the sulfur atoms of the two cysteine amino acids. Okay, so domains are seen in tertiary structures only. So, so domains are basically a combination of one or more secondary structures. Okay, so like you have two alpha helix and one beta sheet, and if it is maintained in the in many proteins, we call it a domain because the heredity is maintained, like alpha beta alpha or beta alpha beta, the beta hairpin. Like you have multiple beta strands, maybe 4 or 5 beta strands, and it has a similar secondary structure that is maintained in the proteins. That is what do I mean by a domain. Okay, now are these domains? How are the tertiary structures formed? One or more secondary structures leading to the formation of a tertiary structure with a proper arrangement followed by stabilizing interactions, right? If that is so, and if that is clear, so domains are maintained in tertiary structures only because a tertiary structure is formed by the different secondary structures. And if a secondary structure is arranged in such a manner that its heredity is maintained, it maintains its if it has it falls in a particular category where that particular secondary structure orientation is maintained in its similar kinds of proteins, then yes, domains are definitely seen in secondary structures only, because in tertiary structures only, because secondary structure is individual, alpha helix beta sheet turns, but say a tertiary structure can be a combination of all of these. That is why here the term domains or motifs play an important role. Okay, if that's clear, then we have the quaternary structures where we have the monomeric proteins and the oligomeric proteins. Now, tertiary structure is where a single molecule of a protein ends, but a single molecule of a protein or a single structure of a protein may not be active. So, we have the oligomeric proteins where that is built by more than one polypeptide chains called the subunits or the monomers. Okay, so one protein molecule or one monomer, the monomeric protein might not be in its active state, so you might need supporting or the subunits of it can be a it can be a homogeneous protein as well, or a heterogeneous protein as well, having multiple types of structure. Like different proteins can come together and make it active. You have the partner proteins that happens in the transcription factors that you see, and also for many enzymes that are not active in its monomeric form but are active in its oligomeric form. That can be a hexamer, a decamer, or a dodecamer. So quaternary structures describes the joining of two or more polypeptide subunits, and these subunits, each of them have their own tertiary structure. So the bonds are the non-covalent interactions over here. So subunits can either function independently or work cooperatively. Like I was mentioning, for most enzymes it happens, like enzymes are in most cases are not active in monomeric form, or it can also happen like enzyme is in its native state likes to be present in an oligomeric form, but when it is in its monomeric form it is active. So it can lead to, in both of the cases, as well as the loss of the subunit or the presence of the subunit can lead to loss in function. Like if you see for an hemoglobin, you have two chains, alpha chain and the beta chain, right? So what happens in case of hemoglobin? So it's a globular protein that consists of four subunits, two alpha and two beta, of two different types, alpha and beta. So each subunit contains a heme group for binding oxygen. Now binding of oxygen to one heme group facilitates the oxygen binding to the other subunits. Now replacement of even one amino acid in the primary structure with another amino acid will hamper the function of the protein. So this is one example where all of these four subunits need to be present for the binding of the oxygen. Now how does it happen? So oxygen binds to one particular subunit that facilitates the interaction of mm, the binding of the oxygen to the other subunits, and in this way the four subunits remains close to one another. That is involved in the function of the protein. So protein structure is hierarchical, hierarchical, where you have the primary structure, that's the sequence of the proteins, followed by the secondary structure which has the local folding based on the types of amino acids. You will have followed by the tertiary structure with the long range folding having the stabilizing interactions between them. Now we have quaternary structures, the multimeric organization, and the large scale assemblies that leads to supramolecular structure formations. Now, once we are aware of the structure of a particular protein, we can also determine the function. We understand that by the protein folding problem, so that determines the structure from where we can get to know about the function. So how can we know about the structure? We have different experimental methods, and there are several computational predictions that now we have got AlphaFold that very well predicts the structure of a particular protein, mostly for folded proteins, though it is not very well able to tell about the intrinsically disordered protein, but for a folded protein it works fantastically. You have several experimental methods like X-ray crystallography, NMR spectroscopy. You have the cryo-EM methods that are very uh, you say it works very well for high molecular weight protein molecules. Okay, and the structure of the different proteins are involved in your regulation, different signaling, transport, movement, catalysis, and other functions that are associated with these enzyme proteins or the different other proteins. Now, the protein folding is like the amino acid sequence of a protein that determines the 3D fold. There are some exceptions, like all proteins can be denatured. Some proteins have multiple conformations. Some proteins need the help of chaperones to fold themselves that play active role in the protein folding. So the function of a protein is determined by its 3D fold. So now the question is, can we predict the 3D fold of a protein given its amino acid sequence? Yes, it is definitely possible. How do we say so? Because alpha fold can very well predict it. We know how the interaction between an alpha helix is formed, how an interaction with the beta sheet is formed. Now, taking all this knowledge and information in consideration, the tool has been designed through which we can determine the structure of a protein molecule given when we know about the sequence of the particular protein. Now there is another thing called the Leventhal paradox that says that, for example, if a small protein of 100 amino acids can assume 3 possible conformations of peptide bonds, so 3 to the power 100, that is about 10 to the power 47 conformations. So the fastest motion is about 10 to 15 seconds. So sampling all the conformations would take you about 5 into 10 to the power 32 seconds, and that's about 10 to the power 25 years. But does it take so much time for a protein to fold? And that is why this Leventhal paradox is still studied today, because it might take a few nanoseconds or even less time for a protein to fold. So how does a protein do that? So the overview of the different energies involved over here. Yeah, because for, like, see, it's the fastest motion. So it's the, you're studying the protein dynamics. So the dynamics of, so the fastest motion dynamics can be in your 10 to 15 seconds. You can take even less than that. That's also fine. But yes, so the fastest motion, what we can imagine is 10 to 15 seconds. That's the fastest motion for if we want to sample all the conformations. So for sampling all these conformations, 10 to 15 seconds is what we would require. So that's an assumption that has been taken. Okay, right. Now, there are four types of non-covalent interactions among the biological molecules in aqueous solvent. Like we have, between the neutral groups, we have the hydrogen bonds. Between peptide bonds, we have it. We have the ionic interactions that leads to attraction as well as repulsion. We have the hydrophobic interactions and the van der Waals interactions. Now we have the hydrophobic effect. It is important for folding because every amino acid participates in the protein folding. Hydrophobic molecules are, so if you talk about the hydrophobic effect, the highest number that you see for each of these adds to the hydrophobicity or the hydrophobic effect in a particular protein. So like it's most for tryptophan, then we have for isoleucine, we have the lowest for arginine, lysine and aspartic. So these are mostly hydrophilic in nature. Okay. Now, so what does this hydrophobic effect tell us? So the conformation of a native protein is marginally stable. There are different conditions from those in a cell that can cause the structural changes in a protein, leading to the loss in the three-dimensional structure that is needed. So that we call as the denaturation, because in the presence of different chemicals, all these interactions are lost. A protein, the disulfide bonds might get reduced. There can be so many chemicals that hamper the native state of a protein, and it leads to denaturation. So in a denatured state, the conformation of a protein need not be completely randomized, and there are a number of physical and chemical agents that can lead to denaturation. Like the classic example is heat that completely denatures a protein, and it opens up the entire protein, giving it a very it giving it an uniform linear shape. And on heating, a protein's conformationally remains intact until an abrupt loss of structure occurs over a relatively narrow temperature range known as the TM. Now, the abruptness of the loss of structure suggests a cooperative process in which a loss of structure in one or more parts of the protein rapidly destabilizes the other parts. So this is a cooperative effect. Also, denaturation can happen due to change in pH, addition of miscible organic solvents like alcohol, acetone. Also, in the presence of reducing agents like guanidium hydrochloride, then urea, the covalent bonds easily breaks up, and it also disrupts the hydrogen bonds, leading to loss of structure. And in the extremities of pH, which alters the net charge, causing to the electrostatic repulsion and the disruption of the hydrogen bonding. So these are very, very important. And the denatured structures results from these various treatments may not be same, but what happens when the, like for the pH, when the pH is very close to the isoelectric point, it leads to aggregation, precipitation of the unfolded proteins. The protein unfolds and it leads to aggregation and precipitation. Like one example is the protein precipitate is seen after boiling of an egg white. That is also a very nice example. Now for renaturation of a protein, now for like we have a denatured protein. Now for renaturation of a protein, there was one very famous example, Aphenson's experiment. Like early studies examined the renaturation of an enzyme ribonuclease, provided experimental proof that the tertiary structure of a protein is determined by its primary structure. In this experiment, the purified ribonuclease was denatured to its unfolded inactive state and was exposed to a solution of urea and the reducing agent beta-mercaptoethanol. Now this unfolded protein and the reduced all the disulfide cross linkages to the cysteine residues. When urea and beta-mercaptoethanol were removed, the randomly coiled denatured ribonuclease spontaneously refolded to its catalytically active state. So this is that there is a minute boundary or a line where there is denaturation and renaturation. In the absence of a denaturating agent, a protein renatures. But like denaturation, the time taken by a for denaturing a protein is very less. It depends on the size of the protein, it depends on the different amino acid orientations of the protein, but how it undergoes renaturation. Okay, like it can undergo denaturation maybe in 10-15 minutes, but for denaturation it can lead to about more than a day or maybe 2 days to get a completely renatured structure of a protein. But if you want to give your protein less time for renaturation, it can lead to random disulfide bond formations. If it does so, it can form random interactions and lead to a crooked structure of a protein. Therefore, slow renaturation of a protein is always recommended. So there is a thermodynamics behind this protein folding. So thermodynamically, the protein folding process can be viewed as a free energy funnel. So the unfolded states are on the top that are characterized by a high degree of conformational entropy and relatively high free energy. Now, as folding proceeds, the funnel becomes narrow, that reflects in the decrease in the conformational space as well as decrease in the entropy. And small depressions along the sides are the semi-stable intermediates. Like you get a folded state, but it can also it can also denature at one point of time or to form a better a folded structure. So you are coming down over here and it's semi-stable state. It wants to again unfold and form a better folded structure, and then you come over here. So these are the semi-stable states. These small depressions are the semi-stable states, and ultimately the end point is your folded structure of a protein. So these funnels can have a variety of shapes depending on the complexity of the protein folding pathway and the existence of the semi-stable intermediates and the potential for the particular intermediates to assemble into aggregates of the misfolded proteins. So ultimately this endpoint is a folded protein. These are the semi-stable intermediates that leads to there is a transition between the folded and the unfolded state, and ultimately the thermodynamics of the protein folding can be explained by this free energy funnel. So that is about the theory of the proteins and protein folding, the denaturation and renaturation. I would give you a 2 minutes break, after which we can start with the practice problems. And in the meantime, if you have any questions, you can put them in the chat. I would be happy to answer. Okay. Okay, so yeah, so let's start with the practice problems. So the first question is, the beginning of the peptide chain has the, so the beginning of the peptide chain has what? Alpha carbon, carboxylic, carbox, carboxylic group, amine group, carbonyl group. A, okay, it has an alpha carbon. Any other answer? So the beginning of a peptide chain or a protein is its N terminus. From where does the name N terminus come? Yeah, absolutely. So it's because of the amino group or the amine group. Yeah, because it's the N terminus. We have the NH2, C alpha, C. That's the backbone. NCC. So the beginning is the amine group or the amino group. That's right. Okay, the next one. A polypeptide chain is generally referred to as primary structure, secondary structure, tertiary structure, quaternary structure. I've got one answer. Primary structure, okay. Anyone else? Yeah. Okay. Where are the others? Please give me an answer. Try to be interactive. Yeah, that's absolutely correct. The polypeptide chain is the primary structure of a protein molecule. The next one. A peptide bond is from the elimination reaction of the carboxyl group, condensation reaction between amine and carboxylic group, substitution reaction of the amine group, addition of H2O to the amine group. B, okay, B, yes, the condensation reaction between the amine and the carboxylic group, the loss of one water molecule. That is correct. Okay. The number of possible tripeptide sequences. What are the number of possible tripeptide sequences? So for example, you have a tripeptide. So what are the possible, or in how many ways can you form a tripeptide? How will you do it? So say you have 20 naturally occurring amino acids. So in how many ways can you form a tripeptide? Yeah, that is good. That's great. 20 into 20 into 20, because I haven't said it cannot be like if in the first place you have alanine, you can have alanine, alanine, alanine. So similarly, for each position you have 20 amino acids. So 20 into 20 into 20, that's about 8,000. Correct. Now the next one. Which of the following cannot denature a protein? You have SDS, sodium dodecyl sulfate, guanidinium hydrochloride, guanidine hydrochloride, iodoacetic acid, and urea. Great. Why? Because guanine hydrochloride and urea are reducing agents. What does SDS do? it opens up the protein because it adds uniform negative charge to all the amino acids, and therefore because of the negative charge interaction, the protein opens up and it denatures the protein. Okay, so now I have a question for you. Like, do you know how many molecules of SDS, how many molecules I mean, how many molecules of SDS will be required to charge, to negatively charge one amino acid? How many SDS molecules will be required to charge one amino acid? So two amino acids gets charged by one SDS. So it's like one SDS molecule can charge two amino acids. Yeah. So it's zero 0.5 actually. That is why I asked. Yeah, no problem. But good try. Yeah. In the peptide bond, the CN bond has what? Only double bond character, only single bond character, both single and double bond character, delocalized electron system. Yeah, good, good. Yeah, actually both C and D are correct. It has a both single and double bond character as well as it has a delocalized electron system. Fantastic answer. The next one, disulfide bond is formed between. Very easy. To cysteine, cysteine serine, to serine, serine and threonine. To cysteine residues. Okay. Now, the phi and psi angles are formed around. This is something out of what I have taught, but I guess you can answer this. The phi and the psi angles are formed around. You have the NH C alpha and C alpha CO, NH CO and C alpha CO, C alpha NH and CO C alpha, the NHC alpha and the NHCO. Yeah, I will be definitely explaining, but before that I want to see if you people know. Okay, if not now, I will be telling in the last because I need to show you a figure for this. Okay, I will show you in the last. Okay, okay. Now next one. I'm keeping this question on hold as of now. I will show you the structure. I will show you the diagram and explaining. Okay. An alpha helix is stabilized by hydrogen bonding, which are formed between the CO NH of ith and I plus 1th residue, C alpha and NH of ith and I plus 4th, CO and NH of ith and I plus 2, CO and NH of ith and I plus 4 residue. Tell me which one. Yeah. What is I? I can be anything. It's a number. So you have 1, 2, 3, 4. It's like that. Yeah. D is the correct option. So ith and I plus 4th residue. So the CO of the oxygen of the ith residue forms a hydrogen bond with the NH, the hydrogen of the NH of I plus 4th residue. Yes, that is correct. Okay, next one. A protein has the following amino acids in its sequence. Which of the following alpha helix are expected to be formed within the above sequence? You have methionine, cysteine, glutamine, arginine, serine, arginine, serine, proline, tyrosine, lysine, glycine, methionine, cysteine, glutamine, glutamine, arginine, serine, proline, tyrosine, lysine. Okay, I got one answer, A and C. Let me see. So the answer is A and C, but let me see what others tell. Now for the time being, you can just answer me, how many residues, minimum number of residues do you actually require for a alpha helix formation? We all know that prolines and glycines are helix breakers. You're not getting the question. I mean, this is the sequence that has been taken from a particular protein. Now the question is, which of the following alpha helixes can be formed, or which of these amino acids can form an alpha helix? That is the question. Or which of the stretch of amino acids can form an alpha helix? That's the question. So if 5 is the minimum number, because you have I and I plus 4, how can a 3 amino acid form an alpha helix? So C is also not correct. Prolines being present cannot form an alpha helix. So only A is the correct option. Right? Next, when a polypeptide folds in an aqueous medium, the loss in entropy is compensated by. When a polypeptide folds, the loss in entropy is compensated by hydrogen bond formation between water and polar residues, loss of water molecules, exposure of polar amino acids to the surface of the protein, release of proton from the acidic amino acids. It's a very direct question. So how is the loss in entropy? Because a polypeptide is actually folding, so its structure is becoming more constant, so there is a loss of entropy, so it has to compensate in some way. How is it compensated? It's a hydrogen bond formation. Okay, you tell an hydrogen bond formation, right? Okay, let's see what others tell. Exposure of polar amino acids on the surface of the protein. So how are you going to compensate the loss in entropy? You mean you have a very, like for example, there are 10 molecules that are randomly moving, that that have very high entropy, and you ask 5 of them to be more constrained, to be more rigid. So you need something to maintain that randomness. The hydrogen bond formation of the polar residues with the water molecules or the polar amino acids on the surface of the protein will not be involved in the randomness that has been taken away from the environment through the through the formation of a folded protein. Yes, the release of the water molecules via the formation of the peptide bonds that are released will also come back to the environment, and that will actually compensate for the loss in entropy. Because these water molecules are free, these can move about in randomly. So you have the 5 molecules that were asked to be rigid, were compensated by the 5 water molecules that are lost through this environment, through the protein, and it goes into the system, right? So the release of the free water molecules are playing vital role in the compensation process. Okay. Next, protein denaturation by heat and urea is mainly caused due to rupture of hydrogen bonds only, rupture of hydrogen bonds and destabilization of hydrophobic interaction, displacement of the water molecules around the protein, or all of these. Yeah. If you see the first one, rupture of the hydrogen bonds do occur because of a reducing agent. Rupture of hydrogen bonds and destabilization of the hydrophobic interactions because of the heat that's produced. Because of the heat, there will be displacement of the water molecules around the protein. So all of these are the factors. Next one. Glacial acetic acid is added to 2 proteins. Protein X of Pi9, protein Y of Pi4 in equal amounts. Initially, both proteins are at pH 7. Which of the following observation is expected to be observed? Protein X unfolds at a slower rate compared to Y. Protein X and Y unfolds at the same rate. Protein Y unfolds at a slower rate compared to protein X. Protein X is affected by acid but not protein Y. Okay, let me try to explain you this. Maybe it might be a little bit tricky. If you can definitely try, but let me try explaining this to you. So what happens, you see one protein has a pi of 9 and another protein is a pi of 4. So one protein we can tell is of a more basic in nature and the other protein is more acidic in nature. Now at lower pH, what happens, the amino acids tend to get protonated more and this affects the structure of the protein to some extent. So both the proteins will be affected when we add glacial acetic acid to it. Now in protein X, it mainly has basic amino acid residues because of its high pi value, which are protonated in nature. At lower pH, since the other residues in X will also get protonated, the net intramolecular repulsion due to similar charges will increase, right? And protein X, therefore, due to similar charge intramolecular repulsion, the protein X will unfold faster compared to Y because the the protonation uh, because it's at a, you say, so what did I say, just let me try to tell you. So protein X has basic amino acids which are protonated, right? So at lower pH, the other residues in X will get protonated. The net intramolecular repulsion due to similar charges will be more, which is not in the case of Y because it has more of acidic residues. So the charge effect does not play a role as much as that in case of X. So therefore, because the intramolecular repulsion is more in case of X, it unfolds at a faster rate compared to Y. Yes, you can also tell in this manner, but C is basically correct. So, yeah. It will also be equal to your pi, so that's also fine. So in any ways, it will start unfolding as well, because it's coming close to a pi, and there will be aggregation, and a protein basically aggregates and precipitates out when it is close to pi. You can answer that in that manner as well, but if you want to answer from the repulsion point of view, you can also tell. But in any way, C is the correct answer. Okay. The next one. Upon adding denaturants with gradual increase in concentration to a protein solution, the absorbance of the protein increases as seen in UV spectrometer. This is due to change in concentration of protein, the unfolding of protein, the gradual exposure of tyrosine and tryptophan residues, the increase in concentration of the denaturant. See, absolutely the gradual exposure of the tyrosine and the tryptophan residues, that is correct. But also the unfolding of the protein, because when the protein unfolds, it leads to the gradual exposure of the tryptophan and the tyrosine residues. Yeah, that's correct. But B also will be the correct option over here. Right, the next one. The temperature at which the structure of a protein breaks is called melting temperature, boiling temperature, saturation temperature, Boyle's temperature. Very easy question. Melting temperature. That is correct answer over here. Okay. Now, now I was left with one question where I guess you only asked me. Yeah. So I will just take out one figure that will help you to. Let me see if I have that one. So this PDF is visible to you. Yeah. Great. So see. So can you see the structure over here? So this is our dipeptide, right? So this is one peptide, and you have this is another peptide, right? So you see there are many angles that are formed. You have the NHC alpha, C alpha CO, C N, N C alpha, C alpha C. Okay, so this is there are different angles across the backbone. So we consider the angles across the backbone, correct? Now, when the angle is between, when we are looking at the angle between NH and the C alpha, it is known as phi. When we are looking between C alpha and carbon, it is psi. These are dihedral angles. So what does that mean? Dihedral angles are ones that are formed between four atoms, right? Like you have a book. For example, you have a book over here. Okay, you open the book across the half, so you have two portions. So in one edge you have one atom. Similarly, you have another atom over here. So if I try to draw, let me see if I can draw. So one atom is over here, one atom is over here, one atom is over here, and the other atom is over here. So it is joined by like this, you go like this and this, and the angle between this is what we want to measure. So for phi, what happens? You have the N over here and you have the C alpha over here. But the other two is basically your CO, C, and this is also your C. Correct? Phi you can also, you call this as phi. Correct? So this is your dihedral angle. Phi is this. Now when the page of the book changes, what happens over here? C alpha comes over here, C comes over here, CO. This becomes your NH, this becomes your this becomes your NH of ith amino acid, this becomes your NH of I plus 1th amino acid, this becomes Chi. So this is what we are seeing over here. So NH, C alpha, phi. However, there are two other amino acids attached. C and C alpha, C alpha and C is Chi, and C and N, that is the peptide bond. The angle across the peptide bond is your omega. So these are the three important dihedral angles present. Now if you, so there the answer is already written, but now if you check, can you answer this, phi and Chi? So the phi one is the phi one is NH C alpha and Chi is C alpha CO. Is it clear to you? Yes, great. Okay, so that's all then. Thank you. I will be sharing the slides with you. Means with the NPTEL team, they would be forwarding it to you, and the video is being recorded, so you can just look back to my YouTube in case you have a problem. Yeah, sure. Go ahead. This one. So what is it telling? So you are adding glacial acetic acid to two different proteins. One protein is basic in nature, the other protein is acidic in nature. How do you explain it? So when a protein is acidic in nature, now when you are adding an acid to a protein, what will happen? The amino acids have will tend to get protonated, right? So the basic amino acids will tend to get protonated, similarly for the acidic amino acids as well. But when a basic amino acid gets protonated, what happens? You have the NH2 that gets protonated, NH3 plus. Now when a protein is more basic in number, what happens? The number of similar charges across one another is more, and when there are you can call intramolecular charge density, the similar charge density very close to one another, the protein will cannot hold back to its native structure and it will unfold. But for an acidic amino acid, these are already protonated, you are adding more of an acid, the protonation rate will be slow. So here we are explaining protonation with denaturation. More protonation across the basic amino acids lead to ionic interaction clash that will lead to the protein X having more basic amino acids to unfold faster. But why being an acidic is being having more number of acidic amino acids, because of its low pi, will undergo slow denaturation rate. Okay. Yeah, Yoshita, you had some query? Yeah, there will be definitely a bit high level because this course is designed for BTechs as well as MTechs as well as PhD. But yeah, IIT Jam is a bit tricky. But yes. So here, whatever the things that I discussed will be there. The questions might be changed in a different manner, but the concepts will cover this portion only. More or less the concepts from this particular protein portion, the structure and types portion will be moreover like this. You can expect many questions from the like a lot of questions will be there from the structures. You can be asked about the pitches of like alpha helix, then the denaturation renaturation thing, protein folding and all. You can be asked. There will be simple questions as well. A very simple question which most of the years they ask is about the structure of the amino acids, and most of the people make a mistake in that. It's not that people skip it, people try to answer it and get negative marking. So try to do whatever has been there. Yes, the level can be a bit here and there, that I'm not very sure. I cannot tell it will be high or low, but the concepts will cover this portion only. Okay. And these exams are basically central examinations. Um, and these exams are central examinations, so it depends on the institute that is setting up the paper and the question setters, the professor setting it. It's very difficult to predict whether how what will be the difficulty level. So I cannot surely tell what will be the difficulty level. Okay, but anyways, so has the IIT Jam? When is the IIT Jam? It's in this month only, or is it over? I don't know. Oh, so you can just, so for IIT Jam, I would be giving you one a small tip, that is whatever you are trying to study, grab the concepts, try to do it throughout how much time you have, do it, but definitely go for at least 20 years of previous year questions. If you get more, that's absolutely fine, but at least 20 years, try for the 20 years. I am definitely going to tell you, even if you only practice the previous year questions of the last 20 years, you're going to crack it very easily. That is a small tip I would just tell you. Try to collect all the concepts and all, but also try practicing the questions. Grabbing concepts and practicing questions are very different. And when you're going to sit for a central for this type of competitive exams, it's very important that you know how to answer the questions. You might know a lot, but if you do not know how to answer the questions, nothing happens. And I've seen a lot of examples like that. So practice the previous years, collect as much information as you can, but also practice the questions. That's all, and best of luck. Okay. Okay, thanks then. Okay, bye bye. We will again meet in the next week.